Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 6, which is the introduction to viruses. So what are viruses? Viruses are non-cellular particle. So this is not living things. Okay, so this is just a particle with a definite size, shape, and chemical composition. This means that they are very specific, so we should be able to identify them based on this characteristic. So the observation of viruses started very early in the scientific development. Louis Pasteur, he postulated that rabies was caused by a living thing smaller than bacteria. Remember, Louis Pasteur was the scientist that uh, postulated the germ theory, promoted the germ theory that infectious disease is caused by germs or microbes. So this is part of his germ theory. Dmitry Ivanovsky, proposed the name for these small particles as virus, mean poison. And then Ivanovsky and Biering showed a disease in tobacco caused by virus. So this is the first observations of the uh, disadvantage of viruses, which is it spoil the tobacco production. The virology itself was a multifaceted discipline. It means that many other disciplines like medical science, plant pathology, maybe science in animal science and animal pathology. They also learned about virus because there are many viruses that can cause disease to whether plant, animal, and even the virus can also cause infection to bacteria. There is no universal agreement on how and when viruses actually originated. Okay, and then virus, because they are very small, they are considered as the most abundant microbes on Earth, and they play important role in evolution of bacteria, archaea, and eukarya. Many scientists believe that virus is actually the bridge between non-living and living things. And viruses are obligate intracellular parasite. Okay, intracellular mean they have to stay inside the cell in order to reproduce. So therefore they are parasite, which is causing harm to the host cell, and they only reproduce. Okay, obligate mean only or must inside the cell, stay inside the cell in order to reproduce. These are some properties of viruses. First, of course, viruses are obligate intracellular parasite. Okay, they can cause problems to the host cell, and they have to be inside the host cell in order to reproduce. The host can be any type of organism, starting from bacteria, protozoa, fungi, algae, plant, animal, including human. Okay? They are very, very small. They are ultra microscopic size. Their measurement is in nanometer, which is 10 to minus 9 meter. It's very, very small unit. Okay, so we cannot use light microscope to see virus. We need the electron microscope to see viruses. They are not cell, so it's not cellular. Okay? So they are very simple and very economical. Okay? So they only have two main molecules as their structure: capsid, the covering, and the nucleic acid in the center. they do not independently fulfill their characteristics of life. So they actually not living thing, but they're able to reproduce inside the cell. So they need living cell in order to fulfill their living characteristic. They are inactive outside the cell. Okay? So it's really easy to destroy the virus outside the cell, but they are very active inside the cell. It is going to be more difficult to control them when they are already inside the host cell. Basic structure only consists of the capsid. Okay, that's the first structure, and then the nucleic acid core. So two main components of virus. Okay, the capsid and nucleic acid core. The nucleic acid can be either DNA or RNA. It cannot be both. It has to be one of these. Okay, so that's the reason why there are two types of viruses based on this nucleic acid: DNA viruses and RNA viruses. The nucleic acid, the DNA, can be double-stranded. This is the most common type of DNA. So remember, it's double helix like this. So this is the most common type of DNA, okay, double-stranded. But there are some viruses that carry very rare single-stranded DNA. So this is rare. And the RNA, okay, the one that carry RNA, mostly will be single-stranded RNA because RNA mostly just single strand. Okay? So single-stranded RNA. But there are some viruses that carry double-stranded RNA, which is again is going to be very rare. The molecules of the virus, okay, in fact, the high specificity. It means that the virus only causing problem or attached to a specific cell. So they causing problem to specific cell or specific tissue or organs. Like for example, hepatitis B viruses is only attached to liver cell and causing problem to liver cell. Flu viruses, they only attach to the respiratory tract mucosal cell and then causing problem to this respiratory tract. So this is respiratory tract infection. So they are very specific. And then they multiply by taking control of the host cell genetic material. They actually hijacking the host cell in order to reproduce. The lack of enzymes, so most of them do not have enzyme, therefore they need to be inside the cell in order to use the host cell enzyme in order to reproduce. They also lack of machinery, so they do not have ribosomes to make protein. So they have to be inside the cell using the host cell ribosome to make their own proteins. The size, again, is very, very small, okay, mostly in nanometer. The biggest virus may be this one, pox viruses. They are about 2 micrometer, but the rest of the virus over here will be very, very small in nanometer. This is maybe the smallest virus over here. It's very, very small. These are okay. These are bacteria. This is Staphylococcus aureus. This is E. coli. Okay, these are two bacteria. This is a coccus bacteria. This is bacillus bacteria. They are bigger, a lot bigger than virus. That's the reason why some virus actually able to kill these bacteria. They can cause infection and destruction to this bacterial cell. Okay, so virus can infect the bacteria. Okay, the structure again very simple. The main molecules are capsid and nucleic acid core, whether DNA or RNA. So that's the two main molecules or components of virus. Okay, so virus particle, the covering, central core. The main covering is capsid. So all virus will have capsid. Okay, so all virus have capsid. Some virus have envelope. Okay, so some have envelope, but not all. Then the nucleic acid, the central cause. Okay, all virus will have this nucleic acid molecule. Okay, whether DNA or RNA. Again, cannot be both. Okay, only DNA or RNA. And some, okay, some will carry enzyme, not all. Okay, some virus has enzyme. Very rare actually, the one that have enzyme. Again, we start with the capsid. Okay, substrate consists of protein is called the capsomers. So this is protein. Okay, so the capsid is actually proteins. And there are some viruses that have envelope. So outside the capsid, there is another membrane that cover the virus. This is what we call the envelope. The virus that have envelope, they also have spike. The spike is the attachment structure of the envelope viruses. And then virus that do not have envelope is called naked. Is naked viruses. The one that have envelope is called the envelope viruses. Okay, so two type of virus based on the outside layer, okay, whether naked without envelope or envelope viruses. Now the capsid, there are two type of capsid, helical or icosahedral. So helical is, the helical is like this, and then icosahedral is mean that it's like crystal with 20 sides. So it's like a crystal a structure. So that's the two type of capsid. So this is the helical type of capsid. So the DNA or RNA will be inside here, covered by this helical capsid. And then this is the icosahedral capsid. It has 20 sides. It's like a crystal, and then the DNA or RNA will be inside this this structure. Now the envelope, again, remember not all viruses have envelope, but if they do have envelope, then they will also have spikes on it. Okay, spike is the attachment structure for the virus that have envelope. Most animal virus have envelope. So animal virus mean virus that attack animal cell, okay, including human cell. So humans considered as an animal. Therefore, the virus that attack human is considered as animal viruses. The functions of capsid and envelope, of course, for protection and also help to bind or attach to the host cell. They also assist for penetrations inside the cell. Okay, there are some non-typical viruses. Okay, remember, typically virus has capsid and nucleic acid. Some of them also have envelope. Okay, naked viruses and envelope viruses. But there are some other viruses, okay, in addition to envelope and capsid and nucleic acid, they have additional structure, like protein and lipid structure over here. This is actually the biggest viruses. This is pox viruses. They have that additional structure. This is non-typical. Another non-typical viruses is the one that attack the bacteria. It's called the bacteriophages. Bacterio mean bacteria, phages mean eater. So this is virus that eat or destroy bacteria. They have special structure over here. They have tail and the head. The tail is special structure that used for attachment. So they have the tail fibers that used for attachment to the bacterial cell. This is the picture under the electron microscope of these bacteriophages because they look like a T letter. Okay? So therefore, sometimes they also call as T phages. Okay, the T phages are the same as bacteriophages. Okay, the genome, the nucleic acid, again, either DNA or RNA, cannot be both. Therefore, based on this nucleic acid, there are two types of viruses, DNA viruses and RNA viruses. The number of genome that they carry is very varies. Okay, it means that there are some viruses maybe carry hundred of genes. There are some only carry 10 or 20 of genes. Okay, there are two types of nucleic acid again, DNA virus, DNA and RNA. Therefore, there are two types of virus based on this nucleic acid, DNA viruses and RNA viruses. Remember, mostly DNA are double-stranded. So mostly this DNA virus will be double-stranded DNA viruses. RNA viruses, mostly they're going to be single-stranded RNA viruses because mostly RNA are single-strand. Okay? There are two types of SS or single-stranded RNA, positive or negative. Positive means they are able to synthesize protein directly using the ribosome of the host cell. So they're able to make their own capsid, they're able to make their own spike, for example, because capsid and spike are protein. So this positive sense RNA able to make the proteins directly. While the negative sense RNA, it has to be converted into positive sense in order to synthesize their protein. So basically, the positive sense RNA will reproduce faster compared to negative sense RNA. If we compare between the NA and RNA viruses, then the RNA viruses mostly will be more dangerous because they are able to reproduce right away in the ribosome because they have the RNA and then they can make proteins right away. They can make capsids, spike. They can also multiply the RNA right away in the cytoplasm. Okay, so therefore they are more dangerous compared to the DNA viruses. DNA viruses, they have to enter into the nucleus in order to replicate. Then they will also produce RNA from their DNA inside the nucleus, and then the RNA will go out from the nucleus to synthesize protein. So the process is longer compared to the RNA viruses. It is the reason why most of the RNA viruses are more dangerous than the DNA viruses. They can destroy the host cell faster compared to the DNA viruses. Okay, so RNA viruses will be directly perform the synthesis in the cytoplasms. They do not need to enter to the nucleus. Okay, so they are faster to reproduce. Okay, the structure of nucleic acids. Okay, so some virus actually carry enzyme. So mostly they do not have enzyme, but there are some virus that carry enzyme. Example is HIV virus. So HIV virus carries specific enzyme. It's called the reverse transcriptase, which is the enzyme that able to synthesize DNA from RNA. Okay, so from RNA into DNA using this reverse transcriptase. Okay, basically, HIV virus is actually RNA viruses. So this is RNA viruses. that able to create DNA, and this DNA will be stay in the host cell, the human host cell, which is the T helper cell. Okay, it stay there. So whenever someone HIV positive will stay HIV positive, because that DNA of virus is actually stay in the host cell. Okay? So this is what we call the reverse transcriptase. Okay? The common process, okay, is actually from DNA to RNA. So this is the common process of uh, transcription. Okay, so DNA usually will be converted or transcribed into RNA using enzyme is called a transcriptase. So transcriptase will be used over here. This is the common process. Uh, so this is what happened in human cell. The DNA will be transcribed into RNA. Then the RNA will be used to make proteins. Right? However, HIV viruses actually have special enzyme that able to convert RNA into DNA, okay? And this DNA will stay inside the host cell. So whenever HIV positive, it will be HIV positive forever. Okay? Classification, because virus has specific size, specific characteristic, they actually can be also classified. But the classification will not follow the organism classification. Okay, they do not have domain, they do not have kingdom, they do not have classes, and etc. So you cannot put them in one of the three domains, them in one of the five kingdom of organism, because they are not living organism. The classification start with the order, okay, usually end with virales, so caudovirales, mononegavirales, nidovirales. So there are three orders. And then under the order, there will be family, usually end with viridae. And under the family, there will be genus and species. And its easy name, they always finish with virus. Okay, so the name usually based on what type of the disease, and then just put virus on it. Okay, like for example, herpes. The virus that can cause herpes is going to be herpes virus. Sometimes we put also human herpes virus. So that's the name, simple name. Okay, the disease, and then the end with virus. Cycle, okay, the stages of viral reproduction. So if we talk about animal viruses, which is the virus that have envelope, okay? So most animal virus has envelope. And remember, if they have envelope, they also will have spike. Okay, so this is envelope viruses. It has 6 stages. Start with the absorption. The absorption using the spike. Okay, so the spikes will be used for absorption or attachment to the host cell. And spike is very specific for specific cell that they bind. Okay. Like for example, COVID-19 spike, they only attach to the cell that's located in, the mucus cell that's located in the nose or in the mouth. That's the reason why the primary transmission of COVID-19 is through the nose or through the mouth. Okay, because they have cells that have receptor for this COVID-19 spike. After absorption, there will be penetration. Okay? So where the virus will enter into the cell. Because they have envelope, then the whole virus will be entered into the cell. Therefore, they need a next stage, okay, which is uncoating. They need to uncoat or remove the capsid in order to release the DNA or RNA. So this is special. Okay, this is special stage for animal virus, which is it has process is called the uncoating. And then after uncoating, the RNA or DNA will start synthesis. If it is DNA viruses, then the DNA, will enter into the nucleus and start synthesis inside the nucleus. If it is RNA, then the RNA will start the protein synthesis in the cytoplasm. Right? And then after synthesis, after they make capsid, they multiply the RNA, DNA, they make the spike, then they will assembly together. So this is the next process, assembly. After the assembly, then they will be exit the cell or release from the cell, mostly using process what we call the budding off or exocytosis. So when they budding off, let's say this is virus inside the cell. Okay, so let's say this is nucleus. They make capsid. Okay, they also multiply their RNA. This is synthesis, and then their assembly over here. The capsid will bind together. Okay, to make this structure to protect and cover the DNA or RNA side. And then they also put the spike on the host cell membrane. So when they're ready to exit, then they exit with process called the budding off, okay, or exocytosis. So this is going to be capsid with DNA or RNA. This is spike, and finally they will exit from the cell as a complete virus with envelope and spike over here. Okay, so this is what we call the exocytosis or budding off from the cell. So this is for animal viruses. Okay, so this is how the process start with the attachment, penetration, then uncoating, remember, and then this is synthesis of their DNA or RNA. This is synthesis of capsid. This is synthesis of spike. The spike then will go outside to the cell, okay, become part of the cell membrane. And then the capsid, nucleic acid will assembly together to make this virus. And then finally it will budding off. When it is budding off, it take the part of the host cell membrane. So this virus over here is actually carry the host cell membrane that has spike on it. Okay, so the envelope is actually part of the host cell membrane. The absorption, as I mentioned before, the spike will absorb or attach to the specific cell that has specific receptor for this specific spike. So it's very, very specific. Like, for example, hepatitis virus only attached to the liver cell because only liver cell have receptor for the hepatitis viruses. Penetration and uncoating. So this penetrate the cell and then uncoating over here. And then replication, assembly, and then release. Okay, so this is assembly, start to the assembly over here, and then finally release by budding off. This is under the electron microscope. You see, several viruses actually exit from the cell. Sometimes when they exit, there are maybe 10,000 viruses will be exit from the cell in one exit, in one release. So the damage that can be caused by the virus is called the cytoplastic effects. Maybe change the cell size and shape. Okay, become bigger. Cytoplasmic inclusion. So when we see under the electron microscope, there will be some extra molecules inside the cytoplasm. And this is what we call the inclusion bodies. or nuclear inclusion bodies. If it is DNA viruses, they will enter into the nucleus. Then there will be extra molecules, is inclusion bodies. Okay? Cell fuse, so some virus actually will attack one cell and then remove the cell membrane, and then the other cell will be also destroyed by the virus, uh, destroy the cell membrane, and then two cells fuse together, become bigger cell. Okay, so virus able to cause some cell fuse together, maybe two cell, maybe three cell, four cell that fuse together finally become one big cell with many nucleus. Cell lysis is mean that virus actually, when it exit from the cell, it will destroy the cell. Like influenza or flu viruses, they perform the cell lysis. They destroy the cells of the host, okay, the host cell. Some also alter DNA, like HIV viruses. They stay in human DNA inside the nucleus, and some transform cell into cancer. Remember, cancer is not infectious disease. This is caused by the cell that divided uncontrollably. Okay. However, some virus actually able to trigger this cancer cell. Like for example, hepatitis B, it can trigger liver cancer. Uh, there are two types of infection actually. The first one is called lytic infection or acute. Okay, lytic or acute. So acute infection is very fast. It destroys the cell within days. Okay? Like for example, Ebola, it can cause lytic or acute infection. It will destroy the cell within several days. Flu virus, for example, they also lytic type of infection. They destroy the some respiratory tract cell right away within days. But type of infection is called a persistent infection, right? Maybe chronic or maybe latent. Okay, so chronic, persistent, or latent infection. The chronic persistent infection is mean that the sign of and symptom of the infection will be observed all day, all the time. Like for example, TB. Okay? Someone that have TB infection, you will be able to see the sign and symptom. Like for example, he's coughing every night. He's gonna have a difficulty to breathe. For example, that's a chronic persistent infection. Sign and symptoms will be present all the time. While the other one is latent infection over here, it means that the sign and symptom only present during outbreak, during special time, like herpes, for example. So herpes virus, they are latent infection. They stay inside the human body, but only give the sign and symptom during outbreak time. Like for example, cold sore, that's a herpes virus. So you only have the sign and symptom of cold sore during certain time, maybe during winter time, for several days. After that, you will not have it again. You will not have it, and then you will have it again maybe next year, etc. That's what we call that latent infection. Again, some virus are able to cause or stimulate cancer. And this virus is called the oncoviruses. Onco mean cancer. Viruses mean virus that trigger the cancer. For example, papilloma viruses, human papillomavirus can trigger cervical cancer. Epstein-Barr virus can trigger the Burkitt's lymphoma cancer. Hepatitis B virus can trigger the liver cancer. Now, the second type of virus is the one that do not have envelope. Right? This is naked viruses. Example is the virus that destroy bacteria. That virus is called bacteriophages. They are naked viruses. Okay, bacteriophages. Mostly they will have what we call the lytic cycle, acute cycle, destroy the cell right away. Now, the stages will be the same, 6 stages, but there are differences over here. Okay, we start with the first one. Of course, it's going to be the same, absorption. But remember, for the envelope virus, they use spike for absorption. This one, the bacteriophage will use the tail fiber, to attach to the bacterial cell. And then the second one is going to be the same, penetration. In this case, the DNA or RNA will be injected out from the virus into the bacterial cell. After that, replication right away. Okay, remember the replication is the same as synthesis. Okay, remember in the animal virus, in the envelope virus, there is uncoating first. There is uncoating. Number 3 is uncoating. But over here, they start the replication or synthesis right away. And then after the synthesis, of course, there will be assembly. And then this is the different over here. Before they release, they need to be mature. So they have maturation. This is only presence in the bacteriophage or virus that do not have envelope. And then after that, they're going to be released. And when they release, they will destroy the cell. So mostly the release is a lysis. It's not budding off. It's not exocytosis, but it is lysis, destroying the cell. Okay, this is the process. Again, start with absorption. Okay, so absorption over here, and then penetration. Okay, so penetration. This is assembly. Synthesis or protein synthesis, and then this is assembly over here, and then maturation, and then release. Okay, when they release, they destroy the cell. You see the one, they actually kill the cell. This is what we call the lytic stage. But the virus, the bacteriophage, also able to go to the persistent infection. The name is not latent, it's not chronic, but the name is going to be lysogenic. For bacteriophage, the persistent infection is called the lysogenic, where they actually will not kill the bacteria, but they will put their DNA as a part of the bacterial DNA. So they're going to stay with the bacteria for a long time. Sometimes, when something happens, they can trigger this into this lytic process. Okay, so they can start, for example, with synthesis, assembly, and then release lytic over here. But they can also stay for longer time in this lysogenic condition. Okay, so again, the process is called the lysogeny, where the virus actually do not kill the bacteria, rather they stay with the bacteria for long term. Okay, so there are two again cycle, going to be acute or lytic cycle and lysogeny cycle. So this is acute, is called lytic. Okay, the persistent is called the lysogeny. This is for bacteriophage. Now, the lysogeny sometimes can make the harmless bacteria become harmful. This is what we call the lysogenic conversion. Example, this one, we're going to use this, okay, Clostridium botulinum. When there is no infection of virus in this bacteria is actually harmless. But if this bacteria has viral infection in it, then this bacteria able to produce toxin is called botulinum toxin or Botox. and this is harmful to human. Especially, it will paralyze human muscle cell. If it is the respiratory muscle, the muscle that used for breathing, then it will kill us, okay, because we are unable to breathe because of this botox. Again, this botox only produced by bacteria that have virus over here, okay, have virus that perform the lysogenic cycle, okay. There are some other examples also for this lysogenic conversion. Okay, so we compare between the bacteriophage, which is this is naked virus, with animal virus, which is it has envelope. This is envelope virus. Okay, so start with the attachment or absorption. It's gonna use tail, right? This is using tail fiber. And for envelope virus, using spike for absorption or attachment. And then after that penetration, usually injecting the DNA, RNA, this one will, the whole virus will enter into the cell. Therefore, after this, after the penetration for animal virus, there will be uncoating. Okay, and you don't see this uncoating in the bacteriophage. It's gonna start with the synthesis and then assembly right away. Okay, for the long-term infection for bacteriophage is called the lysogeny, but for animal virus is called latency or chronic, or sometimes it can cause cancer. Okay. Release from the host cell is going to be mostly immediate or lytic, right away, lytic. But this one is going to be delay, sometimes immediate, like Ebola, for example. Sometimes it's delay, like HIV. Okay, so they usually with the process called budding off or exocytosis. Okay, so that's the difference between animal virus and bacteriophage. Remember, in the bacteriophage, before they release, they will have another process. It's called the maturation. Okay, so these are the difference between animal virus and bacteriophage. Animal virus has uncoating. The bacteriophage has maturation. How to culture the virus? Of course, because virus need living cell, then the culturing method will involve the living cell. Like for example, maybe culture the virus in the cell culture, okay, or in the embryo, like eggs, for example, or inside animal, maybe inside, put virus in the mouse, for example, or rabbit. A prion is smaller molecules than virus because it's only protein, but this protein can cause infectious disease to human. The infectious disease to human is called the Creutzfeldt-Jakob syndrome, or CGS. They can also cause problem to animal like cow. It's called the mad cow disease. So they actually attack mostly brain cell. So the same thing with over here, CGS is a brain cell damage. And prions are extremely resistant to the controlling agent like chemical or radiation, boiling. You cannot destroy prions with that. The other molecule that can cause disease to human is satellite viruses. this is a specific virus that need other virus in order to reproduce. So they need they'll need to work together. So two viruses or three viruses, they combine or they work together in order to be able to replicate. The other one is called the viroid. This is just RNA, particle of RNA that can cause damage to other organisms. So far, viroids mostly identified in plant, so it's causing problem to plants, not animal yet. So this is the other type of molecules, similar or maybe smaller than virus that also able to cause some damages to other organisms, including human. Uh, for treatment, okay, for treatment, uh, when it is outside the cell, virus actually easy to control. So you can wash your hands with the uh, antiseptic soap. You can use alcohol, for example. It will destroy or remove the virus right away. And so it's going to be easy to control virus when it is outside the cell. However, when it is inside the cell, it's going to be more difficult because why? Because they already stay inside the cell. And so if we want to destroy them, usually we also have to destroy the host cell. So that's the reason why the antiviral drugs or medicine usually will cause serious side effects because they actually causing problem to our cell. For example, uh, people with HIV positive, if they take the antiviral drugs for HIV, they usually have a lot of side effects like nausea, vomiting, feeling uncomfortable with their body, and etc. Because the antiviral drug is actually enter into the cell in order to attack the virus inside the cell, and it will cause, of course, some side effects that coming from the entrance of these viral drugs into the host cell. I think that's enough for chapter 6. So we finish with chapter 6. We will meet again with the next chapter, which is chapter 24. See you then. Goodbye.